Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Faiz bin Ismail. In this video, we will be talking about the immune system. Immune system play a crucial role in protecting the body from disease and infection. The immune system is a complex network of cells, tissues and organs that work together to defend the body against harmful microorganisms such as bacteria and viruses. Without a properly functioning immune system, the body would be highly vulnerable to infections and illnesses. The immune system can be divided into two main components, which is the innate immune system and the adaptive immune system. The innate immune system provides immediate and non-specific protection, while the adaptive immune system offers specific and long-lasting immunity after exposure to pathogens. Now we will be talking about innate immunity. Innate immunity provides a broad defenses against infection. Innate immunity is present before any exposure to pathogens and is effective from time of birth. Innate immunity is a non-specific immunity and it provides a rapid response. Next is acquired, or also known as adaptive immunity. Adaptive immunity develops only after exposure to inducing agent such as microbes, toxin or other foreign substance. Acquired or adaptive immunity is very specific. It can distinguish one inducing agent from another. And other than that, adaptive immunity is a slow response compared to innate immunity. Next, we will go to the example for innate immunity. Innate immunity consists of two defenses, which is barrier defenses and internal defenses. For barrier defenses, there are three types of defenses. First, skin, mucous membrane, and secretion. For internal defenses, we have phagocytic cell, natural killer cell, antimicrobial protein, and inflammatory response.
Hi, my name is Muhammad Arif Danish Manzali. Today, I'll talk about the first line of defense of innate immunity. The first line of defense for innate immunity consists three parts, which is skin, mucous membrane, and secretion. First line of defense for innate immunity form the physical barriers that prevent the entry of microorganisms and virus into body tissues. Now, we will move on to the first external defense of innate immunity. The first one is intact skin. Skin is a barrier that cannot be penetrated by bacteria or virus, although even minor abrasions may allow their passage. For the second one is a mucous membrane. It is an epithelial tissue which secretes mucus. It lines many body cavities and tubular organs including the gut and respiratory passages. Certain cells of the mucous membranes produce mucus. Mucus is a viscous fluid that traps microbes and other particles or pathogens. For the last external defense for innate immunity is secretion. Body secretions create an environment that is hostile to many pathogens. It contains lysosome, which is destroy cell walls of susceptible bacteria. It bathes various exposed epithelia by providing washing action that inhibits colonization by fungi and bacteria. In the trachea, ciliated epithelial cells sweep mucus and any entrapped microbes upward, preventing the microbes from entering the lungs. There are three types of secretions for innate immunity. The first one is secretions from the skin, sweat and oil. It gives the skin a pH between 3 and 5, which is acidic enough to prevent colonization of many microbes. The second one is secretion from tears and saliva. It is also include proteins such as lysosome and enzyme that digest the cell walls of many bacteria. The last one is secretion from the stomach, which is stomach acid. The acidic environment which kills pathogen in ingested food or water before they can enter the intestines.
Building on what we've just discussed regarding the body's external barriers, let's move deeper into the internal defenses. The second line of defense against infection is a critical part of our internal protection. This stage depends mainly on a process called phagocytosis. Phagocytes, which are specific types of white blood cells, react to a breach by ingesting invading microorganisms and initiating the inflammatory response. Phagocytic cells, when pathogens defeat the physical barrier defenses of mammals, they are often engulfed by these phagocytic cells. These cells serve to remove cellular debris and respond directly to invasion by foreign substances. They attach to their prey via surface receptors and engulf them, forming a vacuole that then fuses with a lysosome for digestion. The main types of phagocytic cells you should remember are neutrophils and macrophages. To give you an idea of the scale, neutrophils alone make up about 60% to 70% of all white blood cells. They circulate in the blood and are attracted by chemical signals from infected tissue. While they are highly effective at engulfing and destroying pathogens, they tend to self-destruct during the process of destroying foreign invaders. Other key players include monocytes and macrophages, which ingest and destroy invaders and assist in antigen presentation. Eosinophils, which specialize in destroying antibody-coated parasites. Lastly, dendritic cells, which recognize pathogens and activate other immune cells. The actual process of phagocytosis is happen in 6 clear steps. Pseudopodia surround the microbes are engulfed into the cell, a vacuole containing the microbes is formed, the vacuole and lysosome fuse together, toxic compounds and lysosomal enzymes destroy the microbes. Finally, the microbial debris is released by exocytosis. Not all cellular defense is about ingestion. Natural killer or NK cell patrol the body to attack virus-infected body cells and cancer cells. They don't attack the microorganisms directly. Instead, they destroy the infected body cells by triggering apoptosis, which is programmed cell death. This release of chemical inhibits the further spread of the virus or cancer. Our body also uses chemical warfare through antimicrobial proteins. The complement system is a group of proteins in the blood that can activate phagocytes, trigger the release of histamine to start inflammation, or form pores in invading cells to cause lysis or bursting. Interferons are small proteins released by virus-infected cells. They activate macrophage and NK cells and induce nearby uninfected cells to produce substances that inhibit viral replication. Everything we've discussed culminates in the inflammatory response, which is triggered by tissue injury or infection. Its purpose is to limit the spread of pathogens, remove damaged cells, and promote tissue repair. The sequence is as follows. Tissue injury causes mast cell to release histamine. This leads to vasodilation.
So now we will move on to the adaptive immunity. First part of the adaptive immunity, we have antigen. An antigen in any substance that can be specifically recognized by the adaptive immune system and trigger an immune response. Antigens are usually parts of pathogens such as bacteria, viruses, or parasites, but they can also be toxins or foreign molecules. In adaptive immunity, antigens are recognized by B-cell receptors, BCRs, or T-cell receptors, which are also known as TCRs. Each antigen has unique regions called epitopes, which determine how the immune system responds. The specificity of antigen recognition allows the adaptive immune system to target particular pathogen accurately. Now, we will move on to the next part, which is B cell. B cell is a type of lymphocyte responsible for humoral immunity. They develop in the bone marrow and function mainly by producing antibodies. When a B cell recognizes its specific antigen with the help from helper T cells, it becomes activated and differentiated into two parts, which is first, plasma cells, which secrete large amount of antibody. Next is memory B cell, which provide long-term immunity. Antibodies bind to antigen and helps neutralize pathogens, mark them for disruption or prevent them from infecting host cells. The next component is T cells. T cells are a type of lymphocytes involved in cell-mediated immunity. They are mature in the thymus and recognize antigens only when those antigens are present on major histocompatibility complex, which also known as MHC. molecules. There are several major types. First is helper T cells, CD4, coordinate immune response by releasing cytokinesis that activates B cells, macrophage, and other immunity cells. Next, we have cytotoxic T cells, CD8, which directly kill virus-infected cells and cancer cells. Finally, we have regulatory T cells. It is to help prevent excessive immune responses and maintain immune tolerances. T cells are essential for controlling intracellular pathogen and regulating the immune system. For the final component of adaptive immunity, we have lymphocyte development. It is highly processed by which B cells and T cells form, mature, and become capable of recognizing antigens. All lymphocytes originated from hematopoietic stem cells in the bone marrow. B cells mature in the bone marrow, and T cells migrate to the thymus to mature. During development, lymphocytes undergo gene rearrangement to create unique antigen receptors. They also pass through selection process to ensure they can recognize foreign antigens while not attacking the body's own tissues. This process is crucial for immune specificity and cell tolerance.
Next, I want to talk about second immune response. Second immune response occurs when the same antigens enters the body again. Memory B cells and memory T cells formed during the first exposure are rapidly activated. This response is faster, stronger, and more effective, producing a higher amount of antibodies in a shorter time. Because of immunological memory, the pathogen is often eliminated before symptoms appear. Humoral immune response. Humoral immunity involves B lymphocytes and antibodies, and it mainly targets pathogens in body fluids such as blood and lymph. Antigens activate specific B cells. Activated B cells undergo clonal selection. B cell differentiates into plasma cell, secret antibodies, memory B cells, provide long-term immunity. Antibodies bind to antigens, neutralize toxins, and mark pathogens for destruction. Next, we move to cell-mediated immune response. Cell-mediated immunity involves T lymphocytes and is important for destroying virus-infected cells, cancer cells, and transplanted tissues. Helper T cells activate B cells, cytotoxic T cells, and macrophages by releasing cytokines. Cytotoxic T cells or killer T cells directly destroy infected or abnormal body cells by inducing apoptosis. Memory T cells remain in the body and provide long-term protection. Next, we talk about antibodies. There are several functions of antibodies. Antibodies are proteins and produced by plasma cells in response to antigens. There are four functions of antibodies. First, bind specifically to antigens. Number 2, neutralize toxins and viruses. Number 3, promote phagocytosis. And lastly, number 4, activate the complement system. Next, we move to active and passive immunity. For active immunity, it develops when the body produces its own antibodies after exposure to an antigen. It occurs naturally after infection, occurs artificially through vaccination, and long-lasting due to memory cell form. Lastly, passive immunity. Passive immunity is gained when antibodies are transferred from another source. It occurs naturally when antibodies pass from mother to fetus or infant. It can occur artificially also during injection of antibodies. It provides immediate but short-term protection. There's no memory cells are formed.